Hello everyone, my name is Dr. Ashwath Narayan. In this session, we're going to discuss about genome editing techniques. What is genome editing technique? It is a type of advanced biotechnological tools where we can precisely edit the genes whatever we want, and we can insert our gene of interest wherever it is possible. That is called as genome editing techniques. If you observe that, genome editing technique is of three types. One is called as side direction nuclease genome editing technology. Second one is SD1 side direction nuclease, two type of genome editing technology. And third one is directly a GMO, where large scale of DNA elements are edited. So recently, the government of India approved exempted the regulation for the SD1 and SDN2 type of genome editing technology. Why? Because if that will be restricted in India, definitely we cannot achieve a advanced type of techniques. So because of that, the restriction is exempted for SD1 and SD2. But SDN3, there is a restriction for that still in India. Because of that, if you are preparing for UPSC examination, so this is very important that recently in 2022, uh, Indian government exempted for SD1 and SDN2 type of genome editing technology, and then they included that in Environmental Protection Act 1972. And what is genome editing technology? We discussed about, and what is the type of genome editing techniques? In the world currently, many types of genome editing technique is there, like TALEN or CRISPR-Cas9 technology, and other type of technologies are there. In this type of PPT, we're gonna discuss about two type of genome editing technology. One is called CRISPR-Cas9 technology, and second one is talent genome editing technology. First, we're going to discuss about first and foremost genome editing technology called as CRISPR-Cas9 genome editing technology. What is CRISPR-Cas9? First, we need to split up to here what is CRISPR first. So the name itself says that C means clustered, R means regulatory or regularly, and third one is called as I, interspaced. S means short, P means palindromic, R means repeat. The name itself says that clustered. That means a gene of array of DNA is there here. Here, these genes are arranged in clusters. And second one is regularly interspaced. That means the clusters are flanked between some palindromic sequences here. So that is called as regularly interspaced. The interspace, they are flanked in a regular interspace with some palindromic sequence. And then short palindromic repeats. You already know that they have this spacer as well as palindrome both. Because of that, it is called as short palindromic repeats here. And the second part in CRISPR-Cas9 technology is called as Cas9 protein. What is Cas9 protein? Means this is the nuclease protein. This is a dimer, and then it is nucleases. So these nucleases will degrade the DNA or RNA in a particular sequence. Remember, that is called as Cas9. So what is both CRISPR-Cas9 is present in bacteria. So it is a adaptive immune system. What is adaptive immune system? Means whenever some pathogens enters, and particular defense mechanism will occurs against that, and then that will acquire the immunity against that pathogen. That is called as adaptive immunity. Here CRISPR-Cas9 technology is called as adaptive immunity. Why? Because uh, whenever a virus enters, some part of the virus particles will be memorized, and then whenever the same virus enters in second time, definitely this CRISPR-Cas9 work against that, and then neutralizes. And I can observe that the Cas9 generally present in a bacteria called Streptococcus pyogenes that is identified, that is the endonuclease. And you can observe that these CRISPR-Cas9 and then CRISPR and Cas9 work together in adaptive immune system in bacteria. And if you want to take the historical background of CRISPR-Cas9, the two Japanese scientists called as Nakata and Ishino, first they identified these whatever the repeated sequence in bacteria. They identified first in E. coli, Escherichia coli. They identified this CRISPR-Cas9 repeats. That's it. And further, some other researchers worked on that, and they identified that these repeats are not native to bacterium, but it is of different origin. They identified, and further, because of the research, two more scientists worked that this CRISPR-Cas9 technology can be utilized for precise genome editing in eukaryotes as well as prokaryotes. So this is the advanced technique. Because of that, they got. I think they are in a Nobel Prize. They awarded, and two scientists plus Jennifer and Emmanuel. They worked on the CRISPR-Cas9 technology, and then they identified that this technology, which is there in bacteria, can be used also for precise genome editing in prokaryotes as well as eukaryotes. So naturally, this system, CRISPR-Cas9 system contains different components. We're going to discuss that one by one. Okay, na? So first we'll start with this CRISPR-Cas9 machinery. So in that, there is a three component that is naturally present in adaptive immune system. So the first one is called as Cas9. Cas9 means, Cas9 is called as CRISPR-associated nucleases. Cas9 means CRISPR-associated nuclease is called as Cas9. And second part of the this CRISPR-Cas9 technology is called as pre-CRRNA or CRRNA. You can see this is the pre-CRRNA or CRRNA, which is complementary to the virus DNA. Remember, some virus particles enters, so this CRRNA is produced. These are the complementary sequence to the virus genome, that is CRRNA. And one more type of RNA is there, that is flanked to CRRNA, called as tracer RNA. So that is flanked to the pre-CRRNA or CRRNA. Remember, these three components are necessary. So this CRRNA recognize a sequence this upstream to the PAM. You know that what is PAM means? That is called as protospacer adjacent motif. Protospacer adjacent motif. It is a 3 to 4 sequence, or the 3 to 6 sequence DNA that mainly present downstream to the complementary sequence, the CRRNA. Downstream, this PAM is present. Naturally, that mainly helps in defending virus particle. That's it. There is no role other than that. But for editing technology, this Cas9 as a CRISPR-Cas9 machinery is modified in such a way that so these two are joined with the loop, and then these two called as small guide RNA. So pre-CRRNA and tracer RNA together, it is called as small guide RNA, and then we'll put some gene of interest, a chimeric DNA or other DNA will be inserted, and then we modified that. Either precisely we can edit the gene of our interest in prokaryotes as well as eukaryotes. Further, we will discuss about first how this CRISPR-Cas9 defense mechanism works. We need to know completely. First, we will move on to that. Listen here. So, whenever a bacteria, the scientists identify that in bacterial genome, some part of the genome is not native to bacteria. These some part of the genes which is flanked between the palindromic sequence is not at all related to or belongs to bacteria, but they belongs to mobile genetic elements. What are mobile genetic elements? You already know that the name itself says that mobile means they are moving from one genome to other genome. They are moving from one place to other place. They are moving from one organism to other organism. Like that, this is called mobile genetic elements. These mobile genetic elements are mainly from bacteriophages, because bacteriophages will go for a transduction process, generalized, specialized. They will go for lytic and lysogenic life cycle. Because of that, they may be from bacteriophage, or they may be from transposable elements. This is called jumping genes, right? They will jump from one place to other place, called transposable transposons. And even they belongs to plasmids. In bacteria, they know they will go for conjugation, transformation. In that, plasmid will be exchanged between two bacteria. So F plus become donates this plasmid and to the F minus, and then the F minus become F plus. This is like these are called as mobile genetic elements. The spacer, which is flanked between palindromes in bacterial genome, is not at all from the bacteria, but it is from the other origin, either from bacteriophage, either from transposable elements, or either from plasmid. Further, whenever if you see the mechanism of defense CRISPR-Cas9 defense mechanism, whenever virus particles infects and they integrates their genome, not integrates, just insert their genome into the bacterial cytoplasm, what happens? This Cas, cascade is present. Cas 1, 2, 3 up to 9 is present. In that, some subunit of Cas will break the viral genome, and then they will intentionally space between the palindromic repeats of bacteria. Bacteria will have circular DNA, some part will have a palindromic sequence. So in between this palindromic sequence, these Cas subunits, Cas1 and Cas2 will cut the viral DNA and then place in between the palindromic sequence. Further, what happens? Again, this Cas9, Cas2, Cas1 and Cas2, again any other viral particles in enters, genome is enters, definitely again when they will see the common sequence and then place in between the same palindromic repeats. Because of placing that in between palindromic repeats, this CRISPR, this genome becomes a CRISPR array of different virus genomes that flanked between the palindromic repeats. Understand? And then further, what happens? It will forms a CRISPR array. If you see this, this is Cas9 nuclease. So it is a dimer. In two ways it will flanks the DNA and cuts DNA and then edit the DNA or modify the DNA. That will be there. Further, what happens? This viral genome will be inserted and form a CRISPR array. So this CRISPR array further goes for transcription. This CRISPR array, the palindromic repeats placed between spacers. Spacers are the genome that is cutted from the bacteriophage, virus, whatever. The spacers is not native to bacteria, but they originate from the transposable elements, or from the bacteriophage, or from the jumping genes. That is called as mobile genetic elements. Whenever this CRISPR array undergoes transcription, they produces a hybrid of the spacers and palindrome, a sequence that is called as pre-CR mRNA. What is pre-CR mRNA? You already know that this pre-CR mRNA mainly helps in recognition the target viruses. Remember, so this pre-CR RNA will be formed after transcription, and to the complement to that, this Cas9 nuclease will binds here. Some part of the pre-CR RNA will have a palindromic repeats as well as spacer. In palindromic repeats, some unique sequence is there called as PAM. What is this PAM? That is, you already know that protospace adjacent motif. It is of 2 to 6 base pair, and then it is unique with respect to the palindrome, and it helps in recognition of the spacer sequence. What happens? This Cas9 and one more RNA, tracer RNA is there. This tracer RNA and then the Cas9 and then this spacer, this called pre-CR RNA, form a complex, trimolecular complex, And later on what happens, this complex mainly recognize the further viruses infection. This pre-CRRNA, tracer RNA, Cas9 will go splicing here, and then they will be there in cytoplasm waiting for secondary infection. What happens? This is called as effector complex. Pre-CRRNA, tracer RNA, and Cas9, three component together called as effector complex. So this is called as effector complex. This effector complex further undergoes modification, and then they will be there in cytoplasm. Whenever secondary infection happens, same virus bacteriophage infects secondary time, this Cas9, which is complement to the same virus particles, will be activated. This effector complex will be activated and finds the complementary sequence with respect to pre-CRRNA. You already know that pre-CRRNA will have a spacer repeat, spacer that is complementary to the virus particle. Remember. So, and then you here, whenever pre-CRNA and then tracer RNA and then Cas9 form a complex, effector complex, they will wait for the secondary infection. Whenever secondary infection happens, same bacteriophage infects secondary time, they what happens, this Cas9 binds to the region and finds the PAM. You can observe that this PAM, protospace adjacent motif, will be found, and then they will recognize the site where the spacer is present, and then they degrade the entire genetic material of the virus, and this is called as the adaptive immune system. Why it is called as adaptive immune system? Whenever they form effector complex with the formation of a pre-CRRNA or a spacer of the virus origin, definitely whenever secondary time the virus infects, this complex, the effector complex, reaches the virus genetic material, and then precisely break that into species. Further, this will not integrates into plant genome. It will not integrate into plant genome, and then further transduction will not happen. This way, by adopting the spacer, it can defend the infection of the virus particle. This is the actual mechanism of the CRISPR-Cas9 defense mechanism. Understand? So how this defense mechanism works for genome editing technology? So up to this, scientists discover that this is used for adaptive immune system, immunity of the bacteria. But further, this mechanism, how that can be utilized for the precise genome editing technology. So here you can observe that whenever if you observe this kind of system, you can note that we can uh, make a loop for CRRNA and tracer RNA. When you make a loop for the CRRNA, tracer RNA, that becomes a small guide RNA. Remember, the CRNA and then tracer RNA will be joined together with a loop. Whenever loop joins this pre-CRRNA or tracer RNA, they become a small guide RNA. How you prepare small guide RNA? So we'll prepare a CRNA, we'll prepare in the lab itself the CRNA for a target genome. It may be from in the bacteria, or it may belongs to some eukaryotic organism. Take example, somebody is suffering from a lack of insulin. They are suffering from diabetes, right? What should we do? We will prepare a sequence, or we will prepare a CRRNA complementary to the insulin, right? We will prepare a CRNA that is complementary to the complementary to the insulin gene, right? When you produce that and then place it in multiple folds in different places, what happens? The multiple folds of insulin produced whenever you edit the genome of prokaryotes or eukaryotes, even we can produce in prokaryotes as well as eukaryotes the insulin, so that definitely the lack of insulin who are suffering from the diabetes will be gone because of precisely editing the genome of the bacteria or human beings by giving giving the pre-CRRNA or guide RNA directly into the human genome. We can do that, right? So like that we can easily edit who and all suffering from diabetes, who and all suffering from sickle cell anemia, we can edit precisely, and then we can easily modify the mutated genes here. Understand? Further, we'll discuss about it. So what this pre-CRRNA, we'll prepare the CRRNA in our lab itself, and then we'll make a loop of that, and then we'll flank the chimeric DNA, whatever chimeric DNA, the gene of interest called as chimeric DNA will be inserted. So we'll target a specific genes, and then we'll modify it completely. So we'll further move here. And this small guide RNA guides the Cas9 into the target site, target genome, and then we can precisely edit that. Okay. So this is the mechanism where Cas9 and then small guide RNA, which is which has the pre-CRRNA, tracer and pre-CRRNA is flanked with the loop. They become a guide RNA. They will guide to the target site, and then whenever PAM region is there, protospace adjacent motif is there, they will recognize and then splice the target site, and then that Cas9 is a nucleus dimer. It will cuts in both direction, and then it will form a double stranded break. Whenever double stranded break happen, that will go with the repair mechanism. Further, what the repair mechanism, you already know that there is two types of repair mechanism is present. One is that is recognized in bacteria, and second one is that is there in eukaryotes. In bacteria, what happens? Assume that some double stranded and break happen to target site, right? In bacteria, what happens? Bacteria, they will go with homology directed repairs. That is called HDR. What is homology direct repair means? See, we'll recognize, the Cas9 recognize the target site and then splice it. After cutting of double strand DNA, we'll make a solution that contains a donor DNA, a gene that is modified. We'll only prepare it, and then we'll put it in a liquid or a calcium chloride, other type of EDTA or some other type of chemicals. Will add it, and then they will make a soup of that, and we'll dip that in a bacteria that contains Cas9. What happens, through electroporation, DNA enters to the bacterial cytoplasm, and then this DNA will, which is complementary, palindromic sequence are complementary, they will go for homologous repair, and then this donor DNA will be inserted there, and then it becomes chimeric DNA. So this chimeric DNA will produce our gene of interest, and then they will express and produces a protein of our interest. That is there in bacteria. But in case of eukaryotes, you can see in eukaryotes, they will go with two types of repair mechanism: somatic gene therapy or germline gene therapy. Right? Somatic gene therapy means if somebody in somatic cells, they are affected with cancer. They see, some cancers like blood cancer, they are affected. Take example, they have bone marrow is removed, and then in bone marrow cells, whatever is there, we'll took that and then we'll modify it precisely by CRISPR-Cas9 technology. And after editing, we'll confirm the nature of the cell, and then we'll again get inserted in bone marrow, and later on the modified bone marrow cells will produce the healthy cells of WBCs and RBCs. Even with sickle cell anemia, we can produce a healthy RBCs instead of sickle shaped RBCs if you modify that with CRISPR-Cas9 technology. Remember, one more is called germline gene therapy, where germline gene therapy can be done whenever they are suffering from any inherited disease. We have autosomal dominant, autosomal recessive, X-linked dominant, Y-linked inheritance, mitochondrial inheritance are there. Wherever the target genes are there, we'll precisely go and edit that with this CRISPR-Cas9 technology, and then we can edit and modify that. In eukaryotes, we'll have other type of defense mechanism called as these indels. What is indels means? In means insertion, and then del means deletion. Insertion means whatever the gene of interest is there, that is modified with our novel genes. You can see here, whenever break happened, they will go with repair mechanism, and then our gene of interest will be inserted in the place of the precise cut part. So that is called as insertion, right? Deletion means if we don't want the expression of this gene, and we don't want to modify it, and we need to remove the coding part, or we need to remove the coding part or promoter part. So that is called as deletion, where precisely we will delete the nucleotide sequence, and we'll modify the expression, or we'll completely nullify the expression. This is the way we can easily edit and then modify the gene of interest. This is the CRISPR-Cas9 type of genome editing technology. We know how this CRISPR-Cas9 technology works as a adaptive defense mechanism in bacteria. The same natural type of adaptive defense mechanism in bacteria can be utilized for precise genome editing technology. So what are the applications of CRISPR-Cas9? What are the applications here? So we can easily knock out the specific gene of our interest. You can see here, whenever the Cas9 dimers recognize it, what happens? You can see they will go and see the PAM here. Whenever PAM is recognized, and this Cas9 will precisely cut the DNA and make a double strand breaks here. You can see they will make a double strand breaks by cutting the DNA, and later on what happens, we can easily knock down, and further repair mechanisms will insert the base space. Whatever base space are inserted may be used to neutralize or shut down the expression of gene. You can see further, whenever that unmatched DNA or some other DNA is inserted, they shut down the expression precisely. That is called knocking out. Next one is, we can easily transport some enzyme sequences easily, definitely. Whenever the small guide RNA is flanked with some chimeric DNA, this chimeric DNA will mainly go and binds, and then repair mechanism works, and we can easily transport some enzyme for the expression here. So yes, take example in case of deaminase. What is deaminase means? Which mutate the specific DNA by replacing the cytosine with thymidine. Whenever cytosine is replaced with thymidine, what happens? The expression of DNA is alters, and then the expression of the protein will be shut down. That is called as the enzyme, the transport of some enzyme activity. Even we can increase the fold of expression of genes here. Take example, if you want to increase the fold of insulin production because of insufficient production of insulin in diabetic patients, we can insert more number of genes by CRISPR-Cas9 technology, and then we can increase the fold of transcription of those genes, so that our body will produce more insulin, which even we can produce some of the genes, like human growth hormones, where if somebody is suffering from dwarfism, they need more growth hormone to develop and produce their more height, right? So because of that, we can also insert those genes by CRISPR-Cas9 technology. And even we can insert say we can use this CRISPR-Cas9 technology for the cancer immunotherapy. What is cancer immunotherapy means? We will modify the immune cells in such a way that they precisely recognize the cancer cells instead of the normal cells, and then the immune cells like T cells and B cells will reach up the only cancer cells, and then they will activate the program cell death or apoptosis, and then kill precisely the cancer cells instead of the normal cells. This is we can achieve through CRISPR-Cas9 technology. And one more mechanism, one more type of the application of CRISPR-Cas9 is, we can also give treatment for the HIV patients. You already know that still now we cannot able to produce the vaccine for the HIV patients. Why? You already know that the epitope or a spike protein of the HIV will change the protein sequence in host to host. In different host of human beings, they will change the spike protein, so because of that, the vaccine only produced from the epitopes or something. Because of that, we cannot easily effectively we cannot produce the vaccine for the bacteria, HIV virus. What we'll do instead of that, what we'll inject this CRISPR-Cas9 technology with different guided RNA, What they will reach up the cell, human cells, and then they modify the human genome in such a way that the HIV genome, whenever the HIV genome want to integrate with the defense cells, they directly go against that and then easily effectively nullify the action of HIV, so that there is no propagation of HIV in different cells here. That happens because of CRISPR-Cas9 technology that is guided with the different types of guide RNAs here. Even we can use it for COVID treatment. Even we can use it for some inherited disease like in autosomal dominant, autosomal recessive, X-linked diseases are there. We can precisely design a pre-cRNA, and then they will bind to the mutated part, and then the normal gene will be edited further, and then the inheritance of the same disease will not happen to next generation. Like that we can go for the inherited treatments also. In plant research, we can also use for the improvement of the crop yield and resistant to different diseases, viral disease, fungal disease, bacterial disease. Like the different diseases can be cope up by using precise genome editing technology, by using CRISPR-Cas9 technology. And even we can study the gene function of different unknown gene sequences. Why? Because in some inheritance, multiple genes are there in causing of many genes, many diseases here. Take example like diabetes is multifactorial. That means environment factors as well as genetic factors work together in the incidence of the disease. Even cancer incidence, there should be inheritance of two mutated genes, one from the parents or one from de novo. Like that, some inheritance will be there. How come that occurrence of the disease will be studied by CRISPR-Cas9 technology here. That is with respect to CRISPR-Cas9. Further, we can also go for disease resistant, herbicide tolerance, and nutritional enhancement, even biofortification. We can do that. Even nutraceuticals. What is nutraceuticals? Means insertion of the chemicals, medicinally important chemicals into the plants, and then if we consume that some vegetable crops that is genetically modified with the nutraceuticals, we'll get immunity, we'll get antibiotics, we'll get other type of defense mechanism. Even we can get nutrients in biofortification program. Like that we can apply this CRISPR-Cas9 technology in different ways here. Plant breeding, functional genomics, like that different applications are there. Further we'll move on to one more genome editing technology called as TALEN. What is TALEN? So this is also a gene editing technique that is there in Xanthomonas type of species. This Xanthomonas species use this TALEN technology to modify the genetic material, right? What happens? This TALEN. What is TALEN means? Transcription activator like effector nucleases. To escape from the nuclease activity, these tail proteins are secreted from the xanthomonas. They look like the a effector protein which transcribe and mimic like a eukaryotic transcriptional factors. Because of that, our nucleus will not act against that, and then easily the genome, whatever the bacterial genome, successfully integrates into plant genome and cause diseases. Remember, so these xanthomonas produces this tail protein or talon type of proteins. They mimic like a eukaryotic transcriptors or activators. These factors, because of that, these nucleases like DNases, RNases will not recognize that, and then effectively the genome of xanthomonas will be, and integrates into the plant genome and causes different diseases in rice, pepper, tomato, and further. This is discovered by Dane Carroll or Dana Carroll from the University of Minnesota. We can observe that he is the person who discovered that in Xanthomonas species. And what is the mechanism that works? We need to split these talon into two different parts. T, A, L, E, N. We'll split it. This is called as tail protein, and this is called as nucleus, exonucleus or endonucleus protein. So both together will perform the gene editing technology. And this tail protein mainly recognize some sequence here. We'll discuss about in further slides here. So what happens, this if you take a tail protein, it's a um, it contains n terminal c terminal domain, and if it has a sequence of 34 nucleotide sequences there. This 34 out of 34 sequence, the 32 sequence is unique, and then it is not variable, and this constant proteins will be there. But in place of 12 and 13, these protein amino acid will changes. This amino acid will changes that mainly holds up the specificity in binding of the nucleic acids here. These proteins, these amino acids will be, varies according to the nucleic acid. We have, ATGC, right? ATGC is there. This ATGC specifically, some proteins will modify, and then this protein only gives out the specific binding of the nucleic acids here. This 1 to 11 will forms a helix like this, and 14 to 34 make one more helix, and then the 12 and 13 make a loop here. Helix, loop, helix structures are formed. This loop is very important, which has the variable amino acids, and then they are specific to some nucleic acids. We'll discuss about it. You can see here, this is the binding code here. When this loop contains Ni, it specifically binds to the adenine nucleic acid. And then whenever this loop contains HD, it specifically binds to the cytosine. When this loop contains Ng amino acid, they will specifically binds to thymidine or thymine. And this N, it contains, loop contains NK, they binds to the guanine. See that, specificity. If you know this specificity, we can modify this protein or amino acid in such a way that we can specifically edit the genes and recognize the genes because of this loop formation. Helix and loop, or helix loop helix. This loop is very important, which has variable domain. This has two different types of amino acids are there. They are specifically binds to some nucleic acids here: adenine, cytosine, thymine, guanine. So if we know the sequence, we can easily target the sequence by precisely edit this protein, the tail protein, in such a way that we can easily specifically binds to different types of nucleic acids here. So if you take the crystal structure of this tail protein, you can observe that these this surrounds the bDNA groups in such a way that they can easily bind and edit the genome here. And how this TALEN type of technology works in genome editing. Take example, tail protein is different domain, and then they flanked with a exonuclease or endonuclease. So if you flank that with any exo or endonuclease, definitely it will take part in splicing of precise genes here. So this tail protein will design it, which is specifically, we already know that loop, helix, loop, helix. This loop that 1213 is within your hand. We'll modify it so that specifically binds to ATGC. Right? We'll modify it. If you know the sequence, we'll modify the protein in such a way that precisely binds to some sequences here. Whenever tail protein binds here, we'll attach that with some endonuclease here. This is fork one type of endonuclease, that is type 2 S type of restriction endonuclease. What is the role of this? Means, this fork one mainly recognize away from the recognition site and make a break out of it, and then edit or modify the gene of our interest here. Remember, after breaking of this, what happens, the DNA, if it is a bacteria, they will go for repair mechanism, different repair mechanism. In bacteria, homologous repair. In eukaryotes, will have a non-homologous indel repairs are there. Like that, homologous, non-homologous repair mechanism will change the DNA expression, and then we precisely edit the gene of our interest by TIL technology. As we mentioned, this endonuclease, restriction endonuclease, fork 1 is type 2s type of endonuclease, where you can see this is the recognition state and this is the breaking state, stage here, breaking place here. It is go it is splice the DNA away from the active site or recognition site here. And how this editing technology works here, you already know that specifically this 12 and 13 domain will works and recognition of the DNA. After recognition, this nuclease will break that specific part here. Whenever breakage happens, it will go with repair mechanism. This repair mechanism definitely helps in insertion of new DNA or editing of the existing mutated DNA into a new DNA. We can repair it very effectively because of the specificity of the tails protein. Remember, right? Because of specificity, we can easily recognize the sequence of the DNA, and then whenever you flank the tails protein with the any restriction into nucleus, it will cut a staggered cut or blunt end or other cuts, and then they modify the gene of our interest here. Gene of our interest. Further, if we move with the technology, so it will make a double stranded breaks, and then we can insert the DNA or we can modify the DNA. Insert the DNA is called as knock-in, and then modify the DNA, removing the unwanted DNA is called as knockout mechanism. And then by tails technology, we can easily knock out the DNA. If you take the video, if you see the video, whenever tail protein with the nucleus will recognize the open reading frame, and then what happens, it will modify the DNA, and further repair mechanism will add the DNA, and then the functionality of that DNA will be gone because this mechanism is called knockout. Even we can use this for correcting genes here. Take example, somebody is suffering from sickle cell anemia, where so we need to precisely edit the genes in such a way that it produces dumbbell shape of RBCs instead of the sickle shape. What happens, we can easily recognize that, and then we can modify tail protein in such a way that it will modify it will correct the genes that belongs to sickle cell anemia. And further, because of the modification, semantic gene therapy, the person in bone marrow, they produces normal RBCs instead of the mutated sickle cell RBCs here. Even insert the DNA of our interest, we can easily insert around 300 to 500 base pairs with the affected parts, and we can insert the DNA so that the expression of DNA will be there in multiple folds here. Even we can activate the DNA instead of the nucleases. We'll bind flank the tail protein with the activator. It will activate the expression of genes in multiple folds, and then more number of expression takes place because of the activation. Even we can repress the functionality of the genes by adding repressor to the uh, part here. So that's where we can edit the tail talent. We can use this talent technology in